Okay, thank you everyone. Thank you, Elia, for insisting for this event to occur. This is really not taken for granted, especially in this, under these circumstances, but I do believe that this is how we fight darkness, is by bringing in the light of science and technology. That's what we do here, right? We cherish life, healthy life, long, healthy life. That's what we'll do. Okay, so thank you, everyone. I'm going to talk about the progress made in Minovia, the company that I lead in the past 12 years or so, and our novel strategy to develop therapies in the field of aging. Everyone who knows me knows my passion and where my power comes from. My power comes from mitochondria, as well as yours. And actually, every organ in our body and almost every cell contains these tiny organelles called mitochondria. That are inherited from our mothers exclusively. They contain their own DNA and this encodes about 13 proteins, only 13 proteins, and more than 1,500 proteins are encoded by the nuclear genome targeted to the mitochondria. Evolutionary, these mitochondria are considered to be ancient bacteria that entered into the developing cell, and this is actually the basic of our technology in Minovia. Mitochondrial dysfunction not only causes loss of energy production, but also result in very poor steroid hormone production, non-control of cell death, senescence, telomere elongation, and so on and so forth. All are known as the underlying hallmarks of aging. So we do believe that mitochondria is one of the important hallmarks of aging because energy is, life. And when you have mitochondrial dysfunction, that actually results in multisystemic disorders affecting almost every organ system, as you can see here. But the diversity of diseases starts from rare genetic pediatric diseases all the way until age-related diseases. So how do you solve such a complex and heterogenic disease aspects? We chose to focus on one important organ system, which is the immune system or the hematopoietic system. As we heard today, the immune system plays a very important role in many aspects of health and disease and in aging. One of them, we heard several talks about the importance to remove dead or senescent cells from tissues. And the role of removing these cells relies on the immune system or healthy function of the immune system. So the hematopoietic stem cells reside inside our bone marrows, and they're responsible for the production of all kinds of blood cells. So red blood cells, platelets, and every kind of immune cells. And therefore, healthy and functioning hematopoietic stem cell is important for the function of this organ and immune system. There is a very important role of mitochondrial function in hematopoietic stem cells. Impaired renewal of the HSC cell reservoir in the bone marrow happens when mitochondria is dysfunctional. We can see a shift in the lymphoid and myeloid balance, meaning the body has very hard time fighting infections and removal of diseased or damaged cells from tissues, reduced ability to terminal differentiate to mature cell types. I will show you an example of erythroid in a second that results in anemia, in thrombocytopenia, immune dysfunction, of course, and eventually we do know that all of these cells stuck in organs not being removed cause inflammation, which is one of the hallmarks of aging, of course. In Minovia, what we realized very early in stage is that we can develop therapies to improve mitochondrial function, but it will not show us any benefit if we cannot measure mitochondrial function and dysfunction. So the first thing we did, we invested in developing blood biomarkers to measure the efficacy of our therapies. We are part of a very strong Israel Innovation Authority consortium called the liquid biopsy, where blood samples from patients are received, both healthy control, aged controls, and Alzheimer's disease. There are also some groups working on pancreatic cancers. And the idea is that every group, academic or industry group, are measuring different biomarkers, and then correlations are made between different biomarkers. So we are the group of mitochondria, of course. As you can see here, we have samples received from the clinic from young, healthy controls, old healthy controls, and Alzheimer's disease patients. You can see the ages, diversity. Oh, sorry, I don't know what happened. Sorry about that, but this graph is not apparent. But what you see here, we are actually measuring 3 different biomarkers. One is the complex 1, complex 2 activity in a simple manner with succinate utilization assay. Mitochondrial DNA copy number is a way to measure how many mitochondria there are per cell, and ATP production, of course. This is the energy coin in the cell, that's what's important. When you look at the different biomarkers, you don't see such a big difference. Actually, it's quite obvious that maybe even number of mitochondria are increased in age controls, and that's not surprising because they try to compensate on the loss of activity, perhaps. So the only what you don't see here for some reason is the graph that shows that the ATP is really reduced in between the Alzheimer's disease and the age controls and the young and the Alzheimer's disease control. So that was the significant. But so it seems like it's not that important. However, when you calculate how much energy is produced specifically by the mitochondria per single mitochondrion, that's where you start seeing the significant difference between young and old individuals. So this is a very specific mitochondrial score that we calculate from these 3 parameters. So that means what's the efficiency of ATP production per mitochondrion in the cell. So the highest scores is given to the young population, and all the others are really low. Our positive control is a population of primary mitochondrial disease patients, not surprisingly. So this patient population, this is a Karen-Siren syndrome. This is a deletion in the mitochondrial genome, resulting in a very severe pediatric disease, a deadly pediatric disease. And as you can see, the mito score is very low in these patients relative to the young controls. Alzheimer's disease and old individuals are very similar. The goal is to now try to raise this mito score function using the technology that we are developing. So the focus is on 2 different indications. That's a strategy that Minovia have taken in the past years. We're looking at one side at an ultra-rare pediatric mitochondrial, genetic mitochondrial disease. This is, in our mind, the best model to study age-related mitochondrial dysfunction. And on the other hand, we are looking at an age-related mitochondrial dysfunction. Specifically, we are looking at the bone marrow, as I mentioned, and at bone marrow failure disorders. Pearson syndrome is a disease where there is a significant deletion in the mitochondrial genome, resulting, as I mentioned, in a very severe disorder that causes death in very young age. And the first observation is sideroblastic anemia. These children are very anemic. Their blood precursor cells are not producing red blood cells, and therefore they are blood transfusion dependent. And eventually they develop a multisystemic disease. But the first organ that is harmed is the bone marrow. Very similarly, in an age-related disease called myelodysplastic syndrome, there is also sideroblastic anemia. This is the accumulation of iron around the mitochondria observed in the bone marrow of these cells, resulting again in the lack of ability to differentiate to red blood cells and severe anemia. Those 2 diseases are our target indications. And how are we going to solve them? First of all, we wanted to show that indeed using the mitochondrial scoring, we can identify the myelodysplastic syndrome patients as mitochondrial disease patients. So the mito-score shows that in non-MDS patients, the mito-score is high. In low-risk MDS patients, the mito-score is lower, a little bit higher in high-risk MDS. And eventually, this is, again, the combination of the 3 factors that we are measuring. So myelodysplastic syndrome, age-related disease, is a mitochondrial disease, actually. And what we are proposing is this treatment cycle of mitochondrial augmentation technology, MAT. Our product is called MNV201. We are collecting hematopoietic stem cells by apheresis from the patients after mobilization. And then we are enriching these hematopoietic stem cells with isolated, young, and functional mitochondria. Why young? Because we are using the youngest and healthiest source, which is the placenta, that is only like 9 months old. So that's, in our mind, the best source to use for rejuvenating the hematopoietic system. Those mitochondria are ready to use, off-the-shelf products, cryopreserved, qualified by different methods that we have developed. And eventually, the whole process of enriching the stem cells with mitochondria is a process that takes us 24 hours, and then the cells are washed. Only cells with internalized mitochondria are being infused. Not before we make sure that indeed the mitochondria are inside the cells using our methods. The cells are then infused back to the blood, and they know to home by themselves into the bone marrow and start rejuvenating the bone marrow and the immune system. We have shown before that indeed these mitochondria go into the cells. This is the green labeled mitochondria here in this hematopoietic stem cell, and also that these cells can transfer mitochondria between cells in the bone marrow. So it doesn't end up with just the mitochondria going into the cells outside the body, but also they continue to transfer mitochondria in vivo. We can also use advanced technologies to look at the DNA of the exogenous mitochondria that we use, and we use ataxic, single-cell ataxic, to identify an open chromatin that indicates, of course, genes that are transcribed, but also the mitochondrial genome, because it's a kind of an open chromatin. So we can identify the augmented cell specifically, and about 50% of the cell population is augmented with foreign mitochondria, exogenous mitochondria that have a different sequence. That's how we identify it. And it goes up to about 30% of the population on a single cell manner, okay? But it's very diverse. Not all cells take mitochondria to the same level, and we are trying now to understand, based on the different gene expression, what are the elements that will determine which cell will take up more mitochondria than others. And that is still work in progress. We then show that if we take stem cells from diseased individuals like myelodysplastic syndrome, we show that they have diseased properties. So for example, their ability to differentiate to red blood cells is damaged relative to healthy controls. These are CD34 from healthy donor, and you can see how damaged this ability is. And also their ability to in vivo reconstitute the bone marrow of a mouse is damaged relative to healthy control. And now the question is whether MAT can rescue this damage. So recently, we started 2 different clinical trials with our allogeneic mitochondria product for the first time in humans, and then we get access to patient cells for the first time. And as you can see here, both Pearson syndrome patient cells and myelodysplastic syndrome patient cells show improved ability to differentiate erythrocytes. The most important figure is this. This is the terminal differentiated red blood cells, meaning that mitochondrial augmentation actually improved the ability to terminal differentiate to erythrocytes. That's done in vitro, and then when we inject these cells to a mouse model, this is an NBSGW mouse model that can engraft human cells in the bone marrow, you can see this improved engraftment rate, improved stem cell rate in the bone marrow, and again, improved terminal differentiation of erythrocytes in the bone marrow. And also in the peripheral tissues, you can see that there are a high population of terminal differentiated human cells. Importantly, those are low-risk MDS patients. They do carry a mutation that could cause AML, but they are on the low-risk spectrum. One of the questions we were asked is whether enrichment with mitochondria can cause cancer or can accelerate the progression to cancer. There is no scientific rationale to this because actually cancer cells have reduced mitochondrial activity. However, we said, well, if that's the main question that the regulatory agencies ask and the KOL, we have to answer that question. So first of all, in this mouse, there is a reduction in the mutation load. So don't worry about that. But then we went to the best in class, Dr. Omar Abdelwahab from Memorial Sloan Kettering. He has a mouse model called the NHD13 that has an accelerated aging phenotype, and these mice die at 1 year due to AML. So the MDS progresses to AML. We took the stem cells from these mice, enriched them with a healthy placental mitochondria, and then infused these augmented cells into a healthy mouse that was irradiated. So it has space in the bone marrow to now engraft these augmented cells. When we compare now the ability of these cells to repopulate the bone marrow, you can see what happened to the diseased stem cells. They are actually very poorly producing healthy stem cells in the bone marrow. However, the augmented cells produce very healthy and very comparable almost to wild type. More surprisingly, these cells all produce leukemic cells. This is the CD45 negative cells, while the augmented cells have a delayed development of this AML marker. And that's how the survival curve looks like. Those are the mice that received wild-type cells. Those are the mice who received the diseased cells. They die within 4 weeks. And this is the survival curves of the mice who received mitochondrially augmented cells. So the answer is no, we are not accelerating AML. We are actually delaying AML progression and extending lifespan. And that's the goal. We are using this methodology in patients. This is a summary of a previous product. We are measuring the biomarkers. This is, for example, the healthy copy number of the mitochondrial DNA, showing improvement and increase at 6 months post-treatment. That correlates very nicely with disease severity scores. So they are doing better, and their blood mitochondrial DNA increase. And as I mentioned, we have treated already 7 patients with this program, although we have mice data and in vitro cell data, but we are looking, of course, into these patients' follow-up times to understand whether we are changing their anemia, their need in blood transfusions, and that would be our hint into the future, where we can actually expand from these rare diseases into more common diseases of aging. And we are looking forward to that. As a hint, this is the first patient. We had the longest follow-up for this one. She's doing better in terms of the disease severity scores. The lower is better. She's doing better in terms of her growth. She was going down in her BMI and then stabilized at 6 months, and improvement in her blood biomarkers. This is ATP at 6 months, and this is copy number of mitochondrial DNA at 6 months. So we're very excited about this and looking forward to continue this research, and even submitting for the X Prize, which I think you will find very interesting. And the concept is to do the proof of concept with these biomarkers in these mitochondrial diseases, and then design the trial that would show us improvement in immune function, cognitive function, and muscle function in this design of a trial towards this competition in the next 7 years. Thank you so much for your attention.
Thank you. Questions, please? Yes, you.
Do you expect to use the same types of medications for healthy people against aging?
So healthy people actually do not respond. If I take healthy cells, nothing happens. So it's good also. So there is no toxicity. But the idea is to score your mitochondria, for example, and tell you if you are under the desired score, and then tell you if you require therapy. And then treat, and then measure again after a year or 2 years, and then tell you if you require a second therapy. So the biomarkers are critical here in the definition of who will receive this treatment.
But most people don't do biomarkers.
So that's what
How will you target the population?
we will make sure that those assays become available to the population. There is no assay to determine mitochondrial function, even for patients with mitochondrial diseases. Nothing exists. We will make sure those will be available.
You?
Please talk to the microphone. You said that it doesn't help healthy people, but suppose you have healthy people, but suppose you have an ultra athlete that is facing a challenge like a Ironman race or something like that. Would he benefit from extra mitochondria of his own, or he would not benefit?
So again, we never tried to treat, healthy individuals. That's something the therapy doesn't allow us access into healthy humans because it is a complex cell therapy. It's not like a drug that you take. When we did animal studies on young mice, nothing happened. It means nothing changed physiologically. We didn't test it ultra.
healthy?
Yes, actually, I didn't show it here because of lack of time, but we did try on aged animals, and we do see improvement in cognitive function, muscle function, and kidney transcriptions of genes that are related to mitochondrial function. And that's all we tested. I believe that it's even broader.
We have time for one more question. Yes.
Please pass on the microphones.
So is it actually the incorporation of the mitochondria in different target organs, or is it the ATP levels that it is improved in those organs?
So first of all, I would like to clarify that we are working on the hematopoietic system. And although literature shows that there is transfer of mitochondria to other organs, we didn't find it in significant levels that will justify an improvement in muscle or in brain. But we do see an improvement in muscle and in brain. And we believe this is because of the improvement of energy production by the immune cells and better differentiation to the subtypes of the immune system. So it's mediated through the blood and the immune system, and probably because of better energy production.
Okay, thank you.
Thank you.
Thank you. Okay.